So very good welcome, everyone. My name is Nadine Bongaert. I'm Dutch originally, but I live here now in Paris. I'm doing my PhD in synthetic biology, and I'm also the vice president for Hello Tomorrow, a Paris-based non-profit organization for science entrepreneurship. But today, I'm here to moderate this session about gene editing. With me are Victor Delard and Catherine Bergin to discuss about the latest developments in gene editing. And when I say genome editing, what I actually have to mention as well is CRISPR-Cas or CRISPR-Cas9. And this technology has profoundly accelerated the way that we can now edit the genetic code. Now, you may have heard about CRISPR-Cas on the TV or in newspapers, and it's really representing a breakthrough technology for scientists. CRISPR sounds, as what we've been discussing just before, it sounds a little bit like it could be a brand of a cornflakes, but that's not really the case. What it actually is, biologically speaking at least, is a very primitive form of an immune system. You know, we all have an immune system to defend ourselves against viruses, but the tiny little organisms, bacteria, have a similar kind of immune system. And this is called CRISPR-Cas. When a virus tries to invade a bacterium, what it's actually using is CRISPR-Cas to recognize the viral DNA, and then with this CRISPR-Cas protein, it can recognize the DNA and cut it into little pieces. And by this way, it defends itself from being infected and being killed. So you could see CRISPR as a type of very specific biological scissors that can cut any type of DNA. And not so long ago, scientists began to realize that this CRISPR-Cas could actually be reprogrammed to not only cut the piece of DNA of a virus that tries to infect a bacterium, but in fact that you can use it to edit very precisely any type of DNA from any type of organisms. They've tried it in plants, in mice, also in humans, and it seemed to work in a very universal way. And that's why this whole field of gene editing has really been accelerating in the past few days. So what we would like to discuss today is, where are we with this technology? What are sort of the things that we as normal people can expect from this technology? What are the potential benefits, but also what are the potential risks? And for this, I would like to introduce you to our speakers that we have. So Victor is here with us. He is the founder and CEO of Desktop Genetics. It's a technology-based company in London. And what they're doing is they're using deep learning to provide scientists with software with which they can precisely design and engineer their CRISPR-Cas for their experiments. He holds a master in chemical engineering from the Imperial College of London, and he also did a master in bioscience enterprise from the University of Cambridge. And he is a Cairo Society member and also a nominee for the Forbes 30 Under 30. And next to him is a senior researcher at INSERM. Her name is Catherine Bergin, if I pronounce it well. She's a human geneticist specialized in population genetics, and she studied complex human diseases. But apart from the biology, she also started to get concerned with the socio-economic and the ethical impacts of her research. And therefore, she has started all sorts of interdisciplinary projects with sociologists, anthropologists, and historians to assess the impact of DNA sequencing technologies on human health. She's part of an ethics committee at INSERM, where she has dealt with the ethics around CRISPR, and she's actively participating in public debates around synthetic biology and CRISPR, such as we will have today. So a big welcome to both of you. I would like to begin to ask Victor if you have a microphone. Yeah, okay, you both have. So I briefly explained a little bit what Desktop is doing, but can you maybe explain to us why you started this company and what you would like to accomplish with it, or what you already are accomplishing with it?
Yes, of course. Hi, everybody. It's very nice to be here. My name is Victor. And in answer to your question, Desktop Genetics is a company that was set up really to focus on a particular aspect of CRISPR genome editing. And that aspect is the fact that everybody here in this room is different to their neighbor by about 4 million letters. So the A, C, T, Gs that we so elegantly heard earlier, we have about 4 million differences between each individual out of the total 6 billion that represent our unique genetic code. And so when a technology like CRISPR comes around, and this technology allows us to make these very precise edits, what we're focusing on at Desktop Genetics is applying machine learning algorithms to understand how can we personalize these CRISPR so that they are able to very effectively, precisely and safely engineer one cell type. And then when we move to the next cell type, which is different by 4 million letters, we now adapt the CRISPR and engineer it in a slightly different way to make sure that the precision and the safety and the accuracy of the edit is maintained. And so the company has developed software and algorithms, and we sell products and services for scientists who are doing this in research labs, scientists who are looking at how to translate CRISPR into therapeutic opportunities, and also scientists in the drug discovery world who are looking to use CRISPR to discover better targets.
Great, thank you. This clarifies it a bit more. And Catherine, you are a geneticist, but at one point you moved a little bit more into these interdisciplinary projects, and you got more interested also in CRISPR technology. When did that start? How did that begin for you?
So, hello everybody. So there were several opportunities, in fact. I would mention the first one was that, so you said I'm a member of the INSERM, so National Institute for Research, Medical Research and Health Committee. And we decided as a committee in, I would say, end of 2015 to make a statement, to write a statement on ethical issues around CRISPR, because the technology was making more and more noise. And there was this meeting that was organized in Washington, D.C. by the American Science Society, or sorry, I forget.
Society for Science.
Right, something like that. Oh, well, right. And the British and the Chinese, and they decided to have a meeting to discuss the issue around specifically using CRISPR for modifying human embryos. And so we had this reflection, and we wrote this statement that was more general on the different ethical and social, economical issues around the development and use of CRISPR. That's the first thing. And the other thing is that I've also been, for a couple of years, a board member of a French NGO called Sciences Citoyennes. And with this association, we are part of a broader network of NGOs all around the world, actually, who's interrogating biotechnologies not only from their technological dimension, but from their social and economical impact, given that a technology is never pure technology when it goes in the real world. It always enters the world as it is, which means with economic system and social systems, and we might have the best intentions when we as a scientists develop things, and then when they go to the real world, well, they have impact that we might not have predict. But we might also think of these before releasing and creating and thinking the technology. And the fact is, for biotechnologies, most of the development that have been obtained lately tend to have clear economical impact, in the sense that it is fewer actors that get more profits from the development of these technologies, and that there are many local actors, and that was clear with the development, for instance, of genetically modified plants, that there were many small growers that were actually impacted by the development of new GM crops. Yeah. And so that's an issue, and that we're also thinking, and I've been involved in this way of interrogating social and economic impact of technologies.
Great. So before we really move into sort of all the very complicated ethical aspects of the technology, maybe first let's try to sort of sketch an overview of all the things that are possible. What can we expect from CRISPR-Cas? So without all the things, of course, that play a role in how this technology will eventually enter the consumer market or hospitals, etc. What are some of the things that you see are coming at the moment? What is boiling in the labs?
Yeah, so CRISPR came out in about 2012. The first scientific papers of the researchers came out in 2012. And there is very few times in the world where new biotechnologies make such a rapid advance across the world in so many labs. And so CRISPR was very, very quickly adopted by thousands and thousands of labs around the world, thanks in part to the people who discovered CRISPR, who made it very openly accessible. They made their materials very easy to buy for other researchers so that everybody could get started with the technique. And so what's boiling in the lab? What are people using it for? One of the things that scientists do in the context of human health, for example, is they study patients in a hospital, maybe 1,000, 10,000 patients, to try and find a common genetic link to a disease that they have. So let's say patients have a rare genetic disease, and so we read all these ACTGs from all these patients and study all of that population to try and find, okay, is there a commonality between each of those patients? And then we might discover that, okay, there is this one gene that seems to be broken in all of these patients, and therefore the scientists have a hypothesis. And the hypothesis is, if this gene is broken, then the patient has this disease. And so then the scientists use CRISPR to test this hypothesis. So they take cells that you can grow in the lab, and they use CRISPR to cut this gene and deactivate the gene in the cells, and then see if the cells respond like they do in the patients. And if it does, then they're saying, okay, I'm starting to validate my hypothesis. So then the next step is to take cells from those patients that have the gene that's disrupted, and then use CRISPR to fix it, and see if the cells now behave in a healthy way. And that way they're able to complete the research cycle and say, actually, fantastic, we've now validated the genetic link of this gene to this disease, and we can start to progress with discovering a cure. So that's one of the ways that scientists are using CRISPR. And another way that I think is very exciting, because I have friends who did this in their PhDs, what scientists do also to test some new drugs that they're developing is they engineer animals, like they use mice, for example, in the lab. And if you wanted to create a mouse model of a genetic disease before CRISPR, it would take you about your entire PhD's worth of time, which can be 3 to 6 years for one mouse. And now today we can create these mice in about 3 months. And so scientists are able to get through their experiments at a much, much faster pace. And that's enabled by CRISPR.
Thank you.
Well, I agree that for me, it's clear that it's really useful tool to study the molecular mechanisms underlying life in general, because we've not mentioned it, but CRISPR is actually applicable to any kind of living form of living form, actually, either bacteria, yeast, animal, plants, all kind of living entity can be studied and modified with CRISPR. So it's clearly a very efficient, and because we have been trying to modify DNA for like 30, 40 years, right? It's not, but here it's easier, it's faster, it's cheaper, it's many more people can master the technology without having access to specific labs and specific knowledge. So it can and that's the reason why it has diffused so rapidly. So definitely it is an interesting tool to better understand the biological mechanism of life, and thus of disease, and of producing new forms of modified organism, whatever the organism, right? But my concern is also that because it's so easy, everybody wants to try this, and not fear, but I think there's a big issue that it's turning the study of life towards even always more molecular approach of life, which means that we know that a living phenomena relies on genetics, but genetics is not everything. It's always in an environment, and there's issue of interaction between genes and environment, and there are also issues around environment by itself without, varying with genes. And it's so much easier to study gene effect and DNA effect than environmental effect on an organism. But it's a problem, because then we only have knowledge on the gene effect, but not on the environmental effect. And so the issue with CRISPR is that it pushes us even more towards studying only molecular effects. And so I think as a community we have to be very cautious, not to be too much driven by this kind of facilities brought by technology, because it's a real issue. It's distorting somehow the view we have on living forms.
So yeah, I understand sort of what you're saying basically is we are made up out of DNA, but we're constantly influenced by environmental factors. Something that you didn't mention the term, but epigenetics. So the code on top of the DNA that decides what genes are actually active or inactive. Although I've seen people studying epigenetics as well with CRISPR, so maybe CRISPR can still play a role there, but don't you think that it's just one step in the direction of understanding life, and that we may need new tools, more statistics for the environmental factors and other things, that CRISPR is just a better tool to indeed genetics, and that we can combine it later on?
I'm sure that, it's clearly a new and efficient tool, so I'm just saying that we have to be cautious, because it's a tendency, and CRISPR exacerbates the tendency.
So I've read research papers where, for example, they used CRISPR to specifically correct one of the defected genes. Like you mentioned, you can study it, but you can also put back the right letter that was wrong in a piece of DNA. That works really well for people with sickle cell anemia, people with blindness. I've seen. And then the next question is probably one you're also concerned with. Where does it end? So, what are in your views maybe things that we shouldn't use CRISPR for? Is there what feels right, and where does it go wrong?
Well, I think before that, it's the issue for what it will work, and for what it will not work, actually. Because you mentioned the idea of using CRISPR for curing genetic diseases. And this idea of gene therapy, again, is not new. We have been trying to use gene therapy for more than 30 years now. And, well, we thought it would be easy. We just needed more time and more money. And actually, life is a complex process, and we are far from mastering it and understanding it. So after so many years and money involved in gene therapy, there are only a few individuals in the world that have been cured I'm not sure cured is the right word, because it's been only a few months or years since they have been treated, and it's only for specific diseases that it has been manageable. So what I mean by that, we have to be cautious. Again, I mean, I'm sorry, I'm using this word again and again, but it's not just the technology, even though, again, it's an interesting and powerful one. The issue is not only gene editing, it's also getting to the right cell, modifying the right cell in a human person, and it's being sure that you're not modifying a cell in a lab to produce a new corn or something like that. So you're trying to cure one person at a time, right? So we know there are also oncogenic effects. The person we're trying to save may develop some form of tumors or cancer, right? So it's very picky. So regarding gene therapy, I think we're not yet there.
So in terms of that, because we've not really discussed about it, but it's known that CRISPR has off-target effects. So meaning that CRISPR, you can target a CRISPR to precisely cut in a certain piece of DNA, but it doesn't mean it always cuts exactly there. So I'm very curious to hear from you, Victor, how your software, for example, is trying to solve that in a way.
Indeed, and many people may have seen the recent articles about that CRISPR is not as precise as the previous articles seem to make it out to be. And it means I have a job, because it means that we need to be better at predicting for every CRISPR, where are these side effects, where are these unintended consequences going to happen. And the good news for us is that because it's targeting DNA, and we can load DNA into a computer by sequencing it, by reading those ACTG letters, then we can use software to go and search through those 6 billion letters and use some logic to figure out where we think it's going to cut. And I'd like to say that we've solved it, but we haven't quite yet. But we're getting there, and it's, you know, we're gathering a lot more data that continues to refine the machine learning algorithms through some reinforced learning processes. And it turns out that a lot of the assumptions that we had made back in 2013 and 2014, when we developed the first generation algorithms, aren't quite as true as we anticipated, and that was discovered through more data. One of the tricks, though, is that it's quite hard to also detect these mutations. You have to remember that the way CRISPR works is that if you're targeting a pool of cells, you might have 1 million cells, for example, in a tube, and when you deliver CRISPR to them, then you might have some cells that have a lot of CRISPR in it, some cells that have very little, and when you analyze these side effects, you analyze always over a population. And so sometimes you have some really low frequency events, like 1% or 0.1% or 0.01% that are hard to detect, but those are the ones that may potentially be taking place in a region of the genome that leads to progression of another disease that was unintended. So again, software can be very helpful to address those risks up front and say, we know that with this CRISPR, we have, you know, X, Y, Z% risk of targeting these regions, and therefore, when we modify these cells, these are the facts we have to look for. One of the things that cell therapy does, and there are two forms of cell therapy. There's ex vivo, which is outside of the body, and there's in vivo, where the cell therapy is delivered directly to the patient. And the good thing about ex vivo cell therapy is it allows hospitals and doctors to actually sort of quality control the cells that they have been modified outside of the body before they're given back to the patient. So you have a safety net, if you will, and as long as we know what tests to run on these cells to detect any problems before they go back into the patient, then we can already catch some of these. Obviously not the most ideal solution, but better than introducing this disease right away. Though I would point out that we have many drugs on the market today that have a lot of side effects. And side effects in the context of human therapeutic is always a balance of risk and benefit. Always a balance of risk and benefit. Chemotherapy, and very aggressive chemotherapy, kills all the cells in the body. But we accept that level of risk because the cancer is so progressed that we really don't have any other choice. And so at this point, we accept a higher level of risk of killing cells that are healthy and also introducing mutations in other cells that are healthy as a way to tackle all the cancer cells. And so I think we always have to look at these technologies within that framework of risk and benefit And that way, we can always make sure that the technology is being used for diseases and patients that have the greatest unmet need, where the risk-benefit ratio is a lot more in the favor of, yes, this is a newer technology, but there is no alternative for anybody out there, and therefore people and patients and payers are willing to take that risk. Now, of course, we have regulatory frameworks that, before we even get to that question of risk-benefit, we have tested the safety profile and the efficacy profile of these compounds and these drugs. But at least this provides a way for people to see, okay, there is a path for this to move forward, and we know where we're going to start to focus the development of this technology on the applications and on the indications and diseases that have the greatest unmet need, that have no solution out there.
I agree with what you said, but we also have to broaden then the question, because if we consider, for instance, cystic fibrosis, that's a disease, a monogenic disease, so a disease with a defect in one gene that is responsible for the disease. And so we've been knowing the existence of this mutation in the gene for 30 years now. A few drugs have been developed that work so-so for some specific subcategories of the patients. But still, during over the past 20 years, the mean life expectancy of the patient has been increased by like 20 years because of new ways of taking care of the patients with kinds of specific techniques for lung expectorations and that kind of things, which means that it's true that there might not be alternatives in terms of a molecule of drug treatment, but there are also other ways to take care of person. And I want to stress that, because when we are too much focused on technologies, the risk is that we are investing all of our energy and money in fields that may give someday some results. But there are also all these other less tech and less, maybe flamboyant ways that might be quite efficient, in quicker ways.
I agree, and that's a very good point. But I always think back also to the sort of economic reality sometimes of the world we live in, and that is to say that we can have some very we can innovate on how to manage a disease and how to help increase the life expectancy by having better care of patients, but the people who are paying are always going to be asking the question, at what cost? And that is unfortunately a very real question for the bodies that pay for these treatments. In our system here in Europe, most of that is government funded, and then in other countries it's private insurance. But in the context of at what cost, technologies like CRISPR can provide an alternative way, which is, if they are safe enough and if they are effective enough, can we pay upfront, the one time or two times, however many times it needs to be done, but with the idea of CRISPR being that the change is permanent. So let's say one time an actual fix, and avoid the cost over 20 years or 40 years of patient management. And is that a better investment, even though it's a cash outlay today that may be greater than administering a drug over the lifetime of the patient, means that we don't require treating the patient. So kind of like education, if we invest in education of young children, does that allow us then to save cost on the rest of their life when they are unemployed because they don't have the necessary skills to be able to be employable in the job market? And I think that is always an important economic decision and sort of dimension that needs to be remembered here.
But in terms of economics, so you mentioned it as well, the current gene therapies are extremely expensive. In fact, the first approved European gene therapy just got out of the market because
and if you think
how would that work for because you still need to, what is very labor intensive, I guess, is taking the cells from the patient, re-engineering them, putting them back, monitoring the patient. I can imagine it sounds not as easy as just giving a drug, and actually a pharmaceutical company who can make a lot of money from a patient that has to administer a drug on a regular basis.
Yeah, and that's a side thing. If you there have been a bunch of new drugs that have entered the market recently for some kinds of cancer, for some kind of hepatitis C, and the drugs are so expensive that our French health system is like limit in the terms of its possibility to actually pay for these treatments at the price they are now. So, I mean, the issue of how does it cost to society it's a complex one. So I agree your argument is important, but we really have to take all these possibilities and with a fair economic evaluation, because it's sometimes more complex than just curing or not curing.
It's true. Glybera, the first cell therapy, is retailed at $1.5 million. That's a lot of money when you think about trying to have a drug like that, but for diseases that have incidence rates of hundreds of thousands. So these healthcare systems can't afford that. But you know, at the end of the day, if CRISPR is proven to work and companies develop them for very rare diseases where there is really no alternative, then of course these price tags are going to start off quite expensive. But then companies are going to see this as a market, and that allows people to come and innovate on the process of manufacturing these cells, on the process of lowering the cost so that we can, you know, reach a larger market at the end of the day. I think the other interesting piece, though, is that I see then, so we've been talking a lot about CRISPR as a therapeutic. CRISPR is being used back in the lab as a research tool to help speed up and make it more efficient to bring a drug on the market. Remember, pharmaceutical companies price drugs at a very high price because it takes 10 years from the initial discovery to the bedside of the patient to bring a drug. And it costs, you know, the estimates are between 1 and 10 billion, depending on whether you ask Deloitte or PwC, right? But it costs upwards of $1 billion. And the return on their investment from R&D dollars has been going down and down and down. And so there's a dire need to recuperate that investment, of course, which is why they hike up the price of the drugs. But there's also a dire need of just making the research at the beginning a lot more efficient. And what excites me about CRISPR is how it's making the research at the beginning a lot more efficient, because it's allowing us to target and be a lot more clever about how we target diseases with new drugs, because we understand, and I'm a big proponent of that, we understand the genetics behind the disease, so we know which genes to go after, we know which proteins to go after. And so if you can increase the efficiency of the R&D, then you can hopefully speed up the process. That said, there are many other inefficiencies throughout the pipeline, where you just start tackling each one of them one by one.
Yeah, and one of the reason to explain this